students. It's my big pleasure to introduce this afternoon Professor Edward Trifonov from University of Haifa. What to say about him? There is a story which he told me this afternoon, that if you put his name into the grant, you can be sure that you will not get a grant. So it's only one thing I can tell about him now, and you can see later on. Before Edward will start to speak, I will pass you with our lectures here. So enjoy it. Edward,
thank you. It would make sense before starting to give definitions. When people talking about codes, they often mean completely different things. There is a code of behavior, there is a criminal code, there are all kinds of codes in biosemiotics. What I'm going to talk about is a purely information theory term, the code which is carried by text, in this case by the nucleotide or protein sequences. So the definition of the code would be a sequence pattern or bias responsible for specific biological or biomolecular function. It just happened that I was first to introduce that code in the field of bioinformatics or sequence research, and the definition implies that there are many codes. You all know that the famous translation code called triplet code was spectacularly cracked in 61 by simultaneously 3 different labs of Nuremberg, Ochoa, and Corona. And people are literally have been and still are literally hypnotized by this fundamental discovery. Indeed, it is great discovery, and therefore, to begin with, nobody in his healthy mind would suspect even that there may be some other codes except beyond the translation triplet code. So the first announcement of a second code deciphered appeared in 1988, and this was about interaction between amino acyl tRNA synthetase and tRNA. It's not exactly the code which is written in DNA sequence. It's rather code in broader definition, code of interaction of interesting parties involved. So this was called second genetic code, and occasionally in literature you see that term, second genetic code, in exactly that sense. But 13 years later, another second genetic code has been discovered. That time it was about modifications of histones. And nobody really thought that it is funny that we already have one second genetic code. What about the other one? That pattern perpetuated, and in the year 2006 we got a third second genetic code published in Nature. This time it was about nucleosome positioning. And that is not enough. 2 years later, a fourth second genetic code has been published. This time about specificities of interaction of various transcription factors with DNA. And nobody of the authors, of editors, thought about a previous second genetic codes. And even that was not enough. This year, yet another second genetic code was published in Nature again, and this time about alternative splicing. What it tells you about? The one thing is obvious. There are many codes. Another thing is obvious as well. There is a pathological amnesia in scientific society. Five second codes. 3 of them are published in Nature, 1 in Science, and obviously they couldn't count beyond 2. Birds could count till 7. May I ask ladies permission to take off a jacket? Sorry, I'm old man, I'm with old habits. Once I asked a security officer lady in airport, isn't that disgusting that you have to ask every man to take off the belt? She didn't get the joke. She was very, very serious about her duties. Now, the truth is, there are indeed many codes. Here they are listed. 13 of them could be described in very specific terms. Some of them are already essentially cracked. That is, you could really formulate what is a pattern responsible, sequence pattern responsible for that code. The gene splicing code wasn't really cracked. They even forgot that that code till now has a name Chambon rules, GU and AG. This is all what we know about gene splicing in terms of sequence. Looking at the sequence, you only could say that if you have that GU and AG, then maybe this is an intron. Many of them are still not cracked to the end, and many waiting their turn for explicit description. What are the sequences responsible for those functions? And this is common language about common knowledge about the existence of many codes since 1989. In 1989, the late editor of a journal, Bulletin of Mathematical Biology, Lee Siegel, getting interested in what we are doing, suggested to write a paper for Bulletin of Mathematical Biology. And since I was absolutely sure that if I will write a paper about many codes, that wouldn't be accepted by any journal. And I said that, yeah, I'm ready to write that paper about many codes. And it was published. And I think that in Bulletin of Mathematical Biology, that is the most cited paper. My paper about multiple genetic codes. Now you all know the classical papers by Doolittle, Sapienza, Orgel, and Crick about selfish DNA. I consider that these publications are bordering with crime because they completely misled scientific community so that all the messages carried by the so-called non-coding sequences have been ignored since then, 30 years. Take chimpanzee. Chimpanzee has, in protein coding regions, just a fraction of % different sequences from humans. Where is all the difference between chimps and humans? Not in protein coding regions. All the difference is in protein non-coding regions. And all 30 years until the genome of chimpanzee was published, it didn't occur to anybody except very little group of people that one has to look for other codes. What is there in non-coding sequence which tells chimpanzee from humans? What is all we proud of? Where it is written and how? So I just get you acquainted with few codes, of which the first one, historically, the first code after, sequence code, after the triplet code was genomic code of Bernardi about isochores, that there are regions in the genome which are different by GC content, which has far-going biological consequences. So DNA shape code and later I tell you DNA chromatin code were the next to appear in the discussion. The point is that DNA is not homopolymer. If this would be a double helix with identical elements, then of course you would have in 0 Kelvin, if you know what is that, you know what is that? At absolute 0 temperature, you would have a straight axis molecule. But DNA, being a heteropolymer, even at 0 Kelvin, would not be straight. It would not be deformed, but it wouldn't have a linear shape. The axis of DNA, due to the chemical differences between the various base pairs stacked on the top of one another, would deviate from the straight line, and you get locally even curved trajectory, curvilinear trajectory. When we first thought about terminology, I looked in a dictionary, Russian-English dictionary, and for Kjivoy, I found 2 words, curved and bent. And paradoxically, British or Americans, by same token, they don't make difference between curvilinear by force or curvilinear like that. So when you say bent, that is active term. Everybody understands that you just took something straight and deformed it. But when you say curved, they seem the same. Although often the curved means passive curvature. Like in spectacles you have curved glasses, not bent glasses. Or you have, in the text you have parentheses, curved brackets, they are called, not bent brackets. But it happened that my competitors in the field, Americans, they somehow chose the term bent for this property of DNA to be curvilinear. And it's still dominant in literature, although the dominance slowly going down. Because each time when they say about complex of DNA with protein, in which DNA has curvilinear shape, they have to explain that this shape is due to interaction with protein deformation, and simply to say curved or bent would be not enough for their statements. While in our papers we simply say that DNA is bent. It may well be straight when not bound to protein. Anyway, in Russian, in Czech, in Polish, in German, there are separate terms for actively deformed trajectory and for trajectory which is curvilinear without any constraint. Two different words. So DNA is curved because the base pairs are non-parallel to each other, and you do have two different types of deformation, actually two orthogonal components. Either you have, that is called tilt. Imagine the base pairs as chocolate bars. So they are not parallel, they are deformed like that, or like that. So respectively you have so-called tilt wedge angle and roll wedge angle. Wedge we introduced just to understand it easier. You have parallel base pairs, ideally, then you take a wedge and insert it between them and they become non-parallel. So tilt wedge and roll wedge. And they have been figured out, those wedges, from lots of experiments, calculated from electrophoretic mobility of DNA, or from circularization of DNA. If you take very curved piece of DNA, then you would be able to make a small circle. And then from the diameter of this circle, you could figure out those angles. So the code is doublet code, so that every base pair stack AA2TT has a roll angle here, minus 6.5, opening towards major groove. That's what minus means. And tilt plus 3, opening towards phosphates of AA. It turned out that the largest angle is made by AG, largest roll angle. But interesting point is that you cannot put AG closely together, because next AG would be separated from that one by GA, and GA has opposite direction roll, so they will cancel in some degree one another. A big curvature you get when you put together AAA. They don't cancel each other, and next AA is just about 34 degrees rotated relative to the previous one, and still contributing a lot in the same direction. So this is just one example of strongly curved DNA. If you make a periodical sequence, G, 4A's, 4T's, C, then that sequence, when you run DNA with that sequence in the electrophoretic gel, it runs anomalously slow, which indicates that it is curved. Because in the polymer network of polyacrylamide gel, that DNA could only move head-on through the pores. And if it is like that, then it gets lost each time when it tries to progress. It's very much like a snake. Imagine the poor snake with scoliosis. It would have great difficulties to get through the bushes. Chromatin code. This one is related to the curvature code, to the DNA shape code, the same way as bent DNA related to curved DNA. In other words, these are completely different things. But since both things are with DNA, and DNA has a period 10 point something, then they are both expressed in the sequence periodicity. And because of that, it is often confused. Most of scientific community doesn't know the difference between curved DNA and bent DNA. They think that, if the DNA is curved, then it's all right for nucleosome to form. Wrong. So chromatin code is about the sequence which is responsible for placing the DNA into the nucleosome. In other words, it tells you which sequence is good to place the nucleosome. This is a picture from the lab in Oak Ridge, Tennessee, which was competing with Cambridge Group. Cambridge Group was first to crystallize the nucleosomes. They were next, and they didn't survive, and Cambridge Group did survive. There was another competing group in Baltimore of Ren Mudrenakis, Greek, as you could figure out from the name. So Ren Mudrenakis crystallized histone octamer, that is the protein core of the nucleosome. And that paper was immediately criticized by a letter from Cambridge Group. And that letter said that this was wrong, and that was wrong, and that was wrong, but especially what was wrong is that the Baltimore people, they apparently incorrectly calculated the volumes of the protein molecules in the histone octamer. The correct way, they said, is to approximate the complicated shape of the molecule by ellipsoid or rotation, and then by using the following formula, you could calculate the volume of the particle. The answer of Mudrenakis was very short. No, this is not a correct way to figure out the volume of complicated shaped body. In order to get the volume of complicated shaped body, you model that body and immerse the model in the liquid. Then, according to Archimedes' law, you get exact volume of the molecule. So the history of chromatin code was very complicated, was very bitter, because the trouble is that a chromatin code is a very, very weak message. It could not possibly be strong, because if you make a very strong code for nucleosome formation, you will get very strong nucleosome. And very strong nucleosome in a scale of a whole chromosome will be a big trouble for the cell. In all the template processes, when the nucleosome has to be unfolded, the processes would need lots of extra energy. Therefore, the nucleosomes have to be only marginally stable. Because of that, whatever is the signal telling where the nucleosome should form, that signal has to be very weak. It has to be only a hint where it would be better to form. And then it is a headache for people working with signal processing. Because the signal is almost intentionally very, very weak, as if it is designed for the code crackers not to crack that code. The truth is that nature is not that treacherous. Nature did not design any of its codes to be uncrackable. They are all crackable. but some are very difficult. So the very first indication that there is a code for nucleosome positioning was discovery of about 10.5 base periodicity of some dinucleotides in 1980. These were primarily AAA and TTT dinucleotides, which was later on demonstrated to be a major contributor to the nucleosome positioning. But also you could express it in the form purine, purine, purines, pyrimidine, pyrimidines, pyridines, also periodically repeating. Then many labs, being not satisfied by our calculations, tried their own and got different results. In those pre-genomic studies, when the amount of sequences was very limited, it was very hard to crack the code. The cracking came only when whole genome sequences became available, and especially when the databases of nucleosome sequences became available. First database like that was generated in the lab of Andrew Fire for nucleosomes of C. elegans. It was published in 2006. And since this moment on, I was impatiently waiting when they will publish the final code, because they have all the material to do so. They were not in a hurry. In the meantime, during about 3 years, we developed some collaboration with Andrew Fire, and one day I simply asked him, Andrew, tell me, what are you doing with this database of nucleosome sequences? They cut out the nucleosomes, and they have all those pieces. Where is the code? And the answer was amazing. They have very good computer people there, very strong group. Andrew said, that's the point, Edward, we don't know what to do with that. We don't know how to extract that signal. Now, not to advertise myself, I will have to explain you why that happened. Strong computer people, strong lab knowing perfectly well about everything about chromatin were not able to extract the obvious signal which is right there in front of them. Because the signal is very weak. And it requires a very special training in extracting that signal. What was the special training of Trifonov? When I was in Russia, I of course went through military training. Part of that military training is reflected in the DNA curvature, because those angles, the wedge, roll, and twist, they are all the same angles as of a plane when you calculate the trajectory, or a rocket, a missile, when you calculate the trajectory. So we were very fast in that. But signal processing is another specialty. This was the only course in military training which I did like. And this was when you get on a screen, you get a completely noise, just gray, good for maybe for costume, but not to show that this is a tank. A gray screen. Something sort of a little bit darker in the middle, but what is it? It turns out that signal processing technique, which has been secret in many countries for a long time, which are not in the textbooks, they are able to extract that signal from the screen. After some calculations, you slowly get the shape more and more clear, and at one moment you say, this is a tank, it should not track. And oops, the $1 million rocket is sent to kill that tank. So one day I calculated how many, what is the probability to hit a person of a 7 billion population, all right, 4 billion adults, educated adults. What is the chance to hit the specialist who knows how to extract the weak signal from noise? So you could calculate that probability. All right, then I asked myself, what is the probability to hit the person who knows what are nucleotide sequences in chromatin? You also get that probability. Now, to calculate how many people would know both, you have to multiply these probabilities and multiply it by 4 billion population, and I got a figure number 0.2. And this is me. So after we got that database, we made our calculations, not without difficulties, there were still difficulties, but we made our calculations in about a few months and published a paper. We published 2 papers, one in 2008 by Salih, where he demonstrated that indeed this is a periodical repetition of 5 purines with 5 pyrimidines, 5 purines with 5 pyrimidines. And later on, we got a full sequence, which is CGG, AAA, TTT, CCG, and this is repeating by Idan Gabdank. Interestingly, this is again my reverence to the old tradition. I wrote right away a letter to Andrew Fire. And I said that, listen, we now cracked the code with your fantastic database. I think it would be only fair if somebody of your team would be a co-author in that paper. And I got an answer. Edward, honestly, we didn't partake in this study. The main thing was to crack it. We couldn't. And the database, it is public. It has been public already 3 years. I was astonished to get that answer. Who would refuse to be a co-author over cracking another code? Andrew Fire. Great man, by the way, Nobel Prize winner, in a different thing, in RNA interference. When we got that, we expected actually to get in position of CG, there would be some other dinucleotides. In position of GG, also there would be some other dinucleotides. In other words, the whole code would be rather expressed in form of a matrix. There are 10 positions within one period of bent DNA. And in that position, you have this and that dinucleotide, preferably located. In that position, this, and that. So in other words, this would be a matrix of bendability. But amazingly, we got it in a linear form. If this is in a linear form, and if this is connected to DNA deformation properties, couldn't we figure out this simple linear form much before, just from physics of DNA? And it turns out that it could be figured out very simply just from physics of DNA. We didn't do it long time ago simply because we didn't have enough imagination. We didn't think about such a simple possible linear formulation of the matrix of bendability. So here is the physics. In that piece of DNA, one period long piece of DNA, deformed on the surface of histone-octamer, you have purine elements, purine dinucleotides, which are complementary to pyrimidine dinucleotides, and they should be positioned there because the purines, this is a reading strand, 5'. The purines should be closer to the surface of the histone-octamer because when you deform DNA, you have to unstack interactions between purines and between pyrimidines. So purines are tough. They have strong van der Waals interactions between them. Therefore, it's better to put them down because their deformation is less. Same thing you could say about pyrimidines. Pyrimidines are easier to deform, so put them there, upwards, in this position. So in the reading strand, you would have here purines. Then you go further on, and here you would have pyrimidines. All right? So this is prediction coming from simply economizing on unstacking. To save energy on deformation due to stacking, you better put purines, and pyrimidines periodically. Now, what about strong bases and weak bases? Strong are cytosine and guanine, and weak are A and T. These are base pairs which differently contribute to thermostability of DNA. The same story. If you put them outwards at larger distance from the center of deformation, then you would have large deformation. Deformation here, closer to the surface, would be smaller. So if you want to economize on unstacking due to these different distances, you better put strong ones down. They resist deformation, so better you don't deform them, put them down, while weak ones you better put out. Well, here we have to also involve calculations by Viktor Zhurkin. Actually, he made his first calculations as early as in 1980. And I didn't accept his calculations. These were energy calculations. Energy calculations are always very difficult because you don't know really potential functions. You cannot take into account ions and all kinds of things. So I didn't trust these calculations and simply said that we don't see any periodicity of RYs and YRs along the sequence. AA and TT we do see. Purines, pyrimidines we do see periodically changing along the sequence. But YRs and RYs not. But now when we got that pattern from essentially from sequence analysis from the nucleosomes of C. elegans, a pattern in which CG and AT are exactly where Zhurkin predicted it 30 years ago, I had to admit that he was right. So we incorporated then his energy calculations into the scheme. And now just go and read the sequence. Strong nucleotide, which is pyrimidine. It's cytosine. Strong pyrimidine, cytosine. Strong purine, guanine. Strong purine, guanine. Purine, all right. Weak purine, adenine. Weak purine, adenine. Weak pyrimidine, thymine. Weak pyrimidine, thymine. And you get a whole sequence. Now remember that eukaryotes are AT rich, which means that if you take this sequence, it has only 4 of 10 A's and T's. So more likely the eukaryotes would have that, so that uncertainty with that purine would be resolved in favor of A. And then you get C, G, G, AAA, T, 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 C, C, G. And that is almost exactly the same as we got from the analysis of nucleosomes of fire. This was a strong indication that the pattern, the nucleosome positioning pattern, the code is cracked, because we got it by completely 2 different approaches. One, pure physics, and another, just analysis of experimentally obtained nucleosome DNA sequences. But we didn't stop with that. we noticed that in that sequence CG, AAA, TTT, CG, there are trinucleotides AAA, trinucleotides GIA, GGA, and when we look to the usage of these trinucleotides in the sequences in C. elegans, we got AAA on the top and TTT on the top, ATT, AAT, GAA, TTC, all on the top. What you could make of it? You could make a whole pattern of it. How AA would be continuing to the left? Most likely to another AAA. How that another AAA would continue is not again, if not again AAA. Most likely GAA, because GAA is also on the top of the list of the triplets. GAA continues into TGA. TGA continues into TTG. That one into TTT, TTT. That one is continues to ATT, and so forth. The same thing on the right. AA continues to AAT, ATT continues to TTT, each time taking the most frequent element of the sequence. We finally get that pattern. It is shown below because it could be obtained with different starts. We could start with AAA or we could start with CGA. Because C. elegans indicates that CG is important element. All right, let's start with CG. Starting with CG, during the same procedure, we get that. Interestingly, then when we got it, in the preliminary form I published it, I learned just by chance that this technique has a name. It is Shannon n-gram extension technique. And it was quite exactly the time when we were sending a next paper, deciding where to send it. So we decided to send it to bioinformatics because they will be impressed by Shannon 1948, and indeed they have been very much impressed. If we wouldn't refer to Shannon, that would be never published. So notice that the consensus of this reconstruction is G, AAAA, TTTT, C, which is almost the same what we got before. See? Physics suggests CGG. Sequence analysis of database of nucleosome C. elegans suggests CGR. Shannon suggests CGA. And the consensus of all of this is CGR, AAA, TTT, YCG. This is a final formulation of the chromatin code, which is cracked this year. The database sequence analysis got that pattern a year ago, the sequence analysis, while physics and Shannon have been done this year. So the chromatin code is cracked. Now, the various codes which are present in the sequence, they have one very important and unusual property. They overlap. That is, one and the same piece of sequence could simultaneously encode several different things. I could give you only one example known to me from English text. Take this string and try to read what is written here. Different codes would mean different ways to read. These are different messages written in the same sequence. So here are two things which you could read from here. Either you bring her to the rapist, or you bring her to the therapist. Quite a difference. And there are 6 more combinations which you could make from the same sequence. But this example from a human language is very much artificial example. Human texts are not like that. They're never meant to be read in several different ways. The code of reading human text is only one. But nucleotide sequences and protein sequences, they simultaneously carry many different codes. And I'm going to give you several examples. First of all, how come that they could coexist simultaneously in the same sequence? More often, the sequences are sort of, the codes are sort of punctuated. You don't have a whole sequence busy with one message. A letter here, 2 letters here, 3 letters there, and it all together it makes recognizable sequence for the given code. Chromatin code is very much like that. If you know what you're looking for, say you're looking for a cat in the bushes, you don't have to see a whole cat. You would recognize even when you see only parts of the cat. The same thing in terms of sequences, would be like that. Here on the top is ideal sequence for DNA to place in the nucleosome. CGG, AAA, TTT, CCG, and so on, periodically. So this sequence is on the top. They contain these elements which do match these ones, and therefore if you put that piece of DNA, then due to this deformable element properly positioned, you will get pretty good bending of DNA. Nucleosome is recognized in the sequence. In these cases, it is poor. That sequence and that one have only one element which is suitable for the pattern. But as it turns out, if you have one element in one period and another element in another period and a third element in the third period, then it is already enough to qualify for a good DNA deformable well in the nucleosome. So weak is the signal, so hard it was to extract that signal. Now we're coming to a rather unexpected thing. So we do Shannon n-gram extension, looking for frequency of the trinucleotides. You could do that with dinucleotides, with tetranucleotides, essentially with the same result. But now take prokaryotes. And we did take prokaryotes just for control purposes, because prokaryotes don't have nucleosomes. Oops. Prokaryotes show the same preferential pattern. Wow. The whole thing wrong or what? Indeed, if you continue the same way as with C. elegans, you get in falciparum nucleatum, you get AAA, then followed by TTT, and then something else. In Nostoc, I guess, acquittance, you get those AAAs again, but T only once appearing here. But also if you start with TTTs, you could get full TTTs here and only one A here. But they get to the same consensus. Now, in C. pneumonia, you get GAAA and TTTC. In acetivorans, Micrococcus, I guess, you get a whole picture, GAAA, TTTC, very much like in chromatin. Same thing with Bacillus subtilis. So if you take AT-rich bacteria, they all follow in various degrees a common consensus, GAA, TTT, CCC, that one, which is the same as in eukaryotes. This could be a trivial thing, that we simply went wrong way, and the whole story is wrong. This could be not as trivial thing. Maybe this is a message which is shared by eukaryotes and prokaryotes and has nothing to do with chromatin. Because there could be different messages written on the same sequence using the same sequence elements. So what is that third message? Now we know that prokaryotes, they are primarily protein coding, which means that this sequence should code for something. Let us look what for that sequence would code in prokaryotic proteins. So I put together 3 periods. I split them in triplets, and that is the sequence which you read. Glutamic, asparagine, phenylalanine, arginine, lysine, phenylalanine, serine, lysine, isoleucine, phenylalanine. Then it repeats, because the next after this TTC would be again GAA shown here. So that sequence would be then repeated again. But pay attention now to those amino acids which are here. In red you see non-polar amino acids, phenylalanine, phenylalanine, isoleucine, phenylalanine. In blue you see polar amino acids, glutamic, asparagine, arginine, lysine, serine, lysine. Serine is weakly polar. And you have alternation of polar, non-polar, polar, non-polar. These are the sets of non-polar amino acids, of which our amino acids are isoleucine and phenylalanine. And these are polar, of which our amino acids are arginine, asparagine, glutamic, lysine, and serine. And they go one after another with a period 3.5. What is that period 3.5 in proteins? Strain a bit your memory. Yeah, exactly. It's alpha helix. Well, in textbooks you read that alpha helix has a period 3.6, but nobody puts there plus minus, because it could be anything between 3.3 and 3.8. If you look into crystals, you get that. So this is alpha helix, in which the polar residues repeat every 3.5, every turn of alpha helix. In other words, after the first polar residue you get another one which looks in the same direction, and then another one again in the same direction. Alpha helix with charges on one side and with non-polar residues on another side is called amphipathic alpha helix. It is known since late 70s. Not much studies have been made on amphipathic alpha helices, but we bumped into them. So prokaryotic proteins very often protein coding sequences very often code for amphipathic alpha helices. It has nothing to do with chromatin. It's simply very similar code, letter-wise, but of different nature, telling you different thing. So alpha helices typically are 10-15 residues long, which occupies 30 to 45 bases in DNA. They are often amphipathic, that is, having alternating polar and non-polar residues, with a period 3.5 residues. Now, period 3.5 in proteins, of course, means 3 times that, 10.5 in DNA. Periodicity of DNA in the nucleosome is 10.4. Who cares? It's the same value. In other words, sidedness of alpha helix results in a sidedness of DNA. DNA becomes sided. That is, there is one side in which you bend it and it gets well into the nucleosome. Now, I told you about DNA curvature code. And you remember that the sequence GAAA TTTTC is well curved DNA. Then we found out that the same sequence is good for nucleosome formation. Don't make it simple. It's not bending of DNA in the same direction. As a matter of fact, the curvature of DNA is opposite direction to what you get when you bend it. That sequence, GAATTT, it is curved, but when you try to put it in nucleosome, the best way to do it, it is bend it in opposite direction. The physics of that is not yet clear. But if you remember the sectioned snakes sometimes shown in the market on Sundays, or when you have markets, they are scary snakes like that. They are made of sections, and they could bend easily left, right, but not this direction, right? Now imagine that you make those sections unequal. One side would be one angle, and another side would be another angle. Then it will bend one direction much more than opposite direction. That has to do with that phenomenon. Having a curved DNA, you easier bend it in opposite direction than in the same. Well, let's leave it to energy calculators who would figure out why this phenomenon is observed. And now I draw your attention to the yet another code which is carried by the same sequence. You all know about NF kappa beta transcription factor. It is a very important, probably the most important transcription factor, because it has so many interactions and functions in cancer, in gene regulation, in differentiation, everywhere. It was discovered by David Baltimore. So when you make a collection of the published transcription factors and try to make a consensus of it, you arrive to the consensus GGRAATTYCC. Again, same picture. You have 5 purines, 5 pyrimidines with AATT in the center and with G's and C's at the edges. It's the same as in the nucleosome. Practically the same sequence. So we have the same sequence, or almost the same sequence, coding for DNA curvature, this one, coding for nucleosome positioning, this one, coding for amphipathic helices, both in prokaryotes and eukaryotes, this one, and coding for binding site for NF kappa beta. They all follow the same pattern, GRRAATTYYC. In other words, you have a sequence now which encodes simultaneously 4 completely different, I stress it, completely different messages. And reading only one message, one gets 3 more practically gratis. This reads gratis. So we may conclude now. Not only there are many different codes in the sequences, but also they overlap so that the same letters in the sequence may take part simultaneously in several different messages. And you're talking about domination of triplet code. What are proteins after all? Proteins are instruments. Putting things together, cutting things, all kinds of kinases, transferring things, deaminases. These are instruments. Imagine you coming to a different planet and you see a factory. And you excited, run to which building? Will you run to the building which is responsible for instruments? No, you would be interested in the brain of that factory, in engineering department, what that factory is doing, what it's producing, and not hammer and player or whatever. So proteins, I'm exaggerating, proteins are very non-important. In eukaryotes they take only 3-4% of the genome. All the rest is jammed, literally jammed by many other codes. And we know only just a few of them. So we have to study all those codes. And I'm glad to say that we're not alone now anymore. After 30 years of efforts of cracking the chromatin code, we now have people who seriously say in literature, like Professor Brosius wrote, after all, maybe we shouldn't get lost in all those codes. Just take the definition of Trifonov. And this was the first time I saw my name without reference, just in the text of abstract. So you're welcome to studying new codes. Thank you.
Sure, if you want a short break, or we can continue directly.